and welcome to the WashU Virus Genome Browser. Our browser is a publicly available resource for efficiently visualizing viral sequences. The browser hosts thousands of SARS-CoV-2 sequences from locations all across the globe, in addition to other related viral sequences such as SARS, MERS, and more distantly, Ebola. After selecting a virus species of interest, users can select Data Table, where they are redirected to a sortable table providing all hosted genomic sequences which they can view on the browser. The user can easily navigate the available data by sorting the table by a metadata column of interest, such as country or collection date. If tracks are selected from this table, they will be added to the user's cart. At this point, the user can elect to view their selected strains of interest in the context of all other next-strain sequences hosted by the browser by going to our tree view, or alternatively, the user can view their data in a more traditional browser view by selecting Browser View, which is what we will be exploring here. Upon loading the browser view for SARS-CoV-2, you will notice at the top a panel of drop-down menus for adding data and navigating the genome, as well as settings preferences and a help menu. Beneath that, you can see the entirety of the SARS-CoV-2 reference genome, where green highlighting indicates the portion of the genome that is in your current field of view. Tools to change locations, swap tracks, pan the genome, and zoom in or out are available for easy browsing. When you first load in the SARS-CoV-2 browser, you will see several preloaded tracks in addition to any tracks added to your cart. The first of these preloaded tracks is a Shannon entropy track, measuring sequence diversity across all SARS-CoV-2 strains at each genomic position. The second track shows SARS-CoV-2 RNA expression measured by nanopore direct RNA sequencing performed by Kim et al. 2020. From the same study, the user can visualize recombination events detected by junction-spanning RNA-seq reads and several different RNA modifications, rotating in a dynamic track format. If the user pre-selected any tracks from the data table, they will be loaded below and demonstrate sequence diversity of that particular strain from the reference. White space here indicates a match to the reference, and mismatches, insertions, and deletions are color-coded and can be displayed either by zooming in or selecting the colored bar. Several additional tracks are available to be loaded into view and can be accessed under the Tracks tab. When selecting Annotation Tracks, you can see that the Genomic Ruler and NCBI Gene Locations tracks have already been loaded in. The user can additionally load into view a GC percentages track and/or genome comparison tracks, which compare either MERS or SARS to the SARS-CoV-2 reference. Once loaded in, the comparison track reveals all differences between the two species, which can be best visualized by zooming into a location of interest. As individual research groups continue to study the virus and make their data publicly available, we are actively adding additional data hubs which can also be viewed on our browser. To explore available data hubs, select Tracks and then Public Data Hubs. A list of available data hubs with information regarding their sources, data processing, and display interpretation are available by selecting the collection name. To add tracks from a data hub, you must first select the plus sign on the right. This will populate a facet table below, where you can expand the table based on different metadata terms and select which tracks you would like to add to view. Thank you for watching this introductory video to the WashU Virus Genome Browser. We are actively updating this site and would appreciate any suggestions, including additional datasets to add, bug reports, and comments on our GitHub page. which can be accessed under the Help menu. Further documentation is also available here.